So this is chapter 14, in which we're going to look at the brain and cranial nerves. So we'll start with an introduction to some brain terminology and then introduce you to the major regions of the brain, which we'll go into more detail with later. So the first major part of the brain, and the part that most people think of when they think of the brain, is the cerebrum. So the cerebrum is this large wrinkled part at the top. And if you look at the cross section down here, the cerebrum would be, again, this top wrinkled part. These lower parts are other sections that we're going to talk about soon. And the cerebrum itself is divided into two hemispheres, a right and a left hemisphere. So the picture at the bottom shows a cut that was made straight through the two hemispheres. So now we are only seeing one of the hemispheres here. This would be the right hemisphere. And the outer layer of gray matter in the cerebrum is called the cerebral cortex. And remember that the gray matter is where you find neuronal cell bodies and unmyelinated axons. So the more wrinkly a brain is, the more space you have for gray matter. And it's thought that this does correlate some with intelligence and problem-solving skills and social ability when you compare brains between different species, as we'll look at in a moment. But again, the more wrinkled a brain is, the more extra space you have to have a larger area of cerebral cortex, which again is where the neurons are actually located. So some terminology to know: the gyri are the elevated ridges that are increasing the surface area. So these would be the wrinkles themselves. So think of, like, if each wrinkle represents like a hill and a valley and a hill and a valley. The gyri are the tops of the hills. So they are the wrinkles or the ridges themselves. And a singular one of these would be called a gyrus. So if I were to point to this one right here, I would say that's a gyrus. All of these together are gyri. Sulci are the shallow depressions or valleys between the gyri, and a singular one of these would be called a sulcus. So I'm dragging my mouse through a sulcus right here, and then sulci would be all of them together. You can also see the gyri and sulci in the cross section. You can see where the wrinkles continue, and you've got the valleys or depressions between them. And then really deep grooves are called fissures. So like the groove that separates the left and right hemispheres is a fissure because it's very deep. And I mentioned earlier that the amount of wrinkling, so the amount of gyri and sulci a brain has, correlates with the amount of cerebral cortex it has, which correlates to the amount of neurons it has, which correlates to the amount of processing power the brain has. So to give you an example, this is a brain from a rat. And you can see that its cerebrum is very smooth. You don't see any gyri or sulci, so it doesn't have the extended surface area that the human brain has. And if we look at a dog's brain, now we can start to see some sulci and some gyri. So now we are getting a little bit more wrinkled. So we typically think of dogs as being more intelligent than rats. They've got higher brain processing power. They also are very social. And interestingly, if you look at a dolphin, their brains are actually more wrinkly than ours. So they have an extremely high surface area of their cerebral cortex, which is interesting. We know dolphins are highly intelligent. We know they're highly social. We know they can use tools and teach their young how to use the tools. We know they have names for each other. They even gossip about each other. So dolphins are very highly intelligent. So the major parts of the brain, so the first one we've already talked about, that's the cerebrum, that's the top wrinkly part. We're going to talk about functions a little bit later, so now I'm just introducing you to the major parts. In addition to the cerebrum, we have the cerebellum, which is this smaller wrinkled part at the very back. Cerebellum actually means little brain in Latin, so they thought this would be the big brain, this would be the little brain. Then you also have the diencephalon. The diencephalon is buried deep inside, so it's this pinkish purple mauve color, and it would be located right in the middle of the cerebrum. So it sits up under the cerebrum. And the diencephalon contains the thalamus and the hypothalamus. And again, we're going to talk about these more a little bit later. And then you have the brainstem, which comes down from the diencephalon and then joins up with the spinal cord. And the brainstem contains the midbrain, the pons, and the medulla oblongata. In this section, we'll look at the ventricular system, which includes the ventricles and passageways within the brain. So the ventricles are spaces or cavities within the brain, and they are lined with ependymal cells that you learned in chapter 12 are the neuroglia that make cerebrospinal fluid. And so the ependymal cells are making the CSF, and then the CSF sits or lives inside of the ventricles and the passageways that connect them. So there are 4 major ventricles of the brain. There are 2 lateral ventricles, one in each hemisphere. So you can see these the best in the anterior view. You can see how you have one in the right hemisphere and one in the left hemisphere. You can also see them on a side view, but one of them kind of overlaps with the other. Because you have 2 lateral ventricles, the next ventricle is called the third ventricle. So you can think of the lateral ventricles as being ventricles number 1 and 2. So the third ventricle itself is located in the diencephalon. So it sits right above the hypothalamus and between the 2 thalami on either side of the diencephalon area. So that's right here in the middle. So this would be the third ventricle. The third ventricle does have a hole in the middle of it, and that is where one thalamus connects with the other. So the left and right thalami connect through this hole. And if you look at the anterior view, the third ventricle is right at the very center midline of the brain. And then the fourth space is called the fourth ventricle. And the fourth ventricle is located between the pons and the cerebellum. So this is going to be down in the brainstem region. So you can see the fourth ventricle here. It's very broad when you look at the anterior view. When you look at the lateral view, it looks more like a passageway, but it's still large enough to be considered a ventricle. And it extends downward. It's between the pons and the cerebellum, but it also extends downward behind the medulla oblongata as well. So those are the ventricles, and then you've got the passageways that connect one ventricle to another, which ensures a continuous flow of CSF throughout the entire ventricular system. So first we have the interventricular foramina, which connect the lateral ventricles to the third ventricle. So they would be the little connecting passageway between the lateral and third ventricles. And this picture can kind of help you see it a little bit better because it's been highlighted in red. So this would be our third ventricle in the middle, our 2 lateral ventricles in either hemisphere, and the interventricular foramen is going to connect those 2. The cerebral aqueduct is the passageway between the third ventricle and fourth ventricle. So you can see the third ventricle here, the fourth ventricle here. The cerebral aqueduct would be the connection between those 2 ventricles. And here is an image where the cerebral aqueduct has been colored green. So you can see how it is stretching between the third and fourth ventricles. And then the fourth ventricle narrows at the bottom, at the behind the medulla oblongata, and it becomes the central canal of the spinal cord, which we'll talk about in chapter 13. And this is going to carry CSF down through the spinal cord. In this section, we'll look at the cranial meninges. So the cranial meninges are continuous with the spinal meninges, which are covered in chapter 13. So both the brain and the spinal cord have very similar layers to their meninges, but there are some key differences. So the outermost layer is called the dura mater, which in Latin actually means tough mother. So mater is mother in Latin, and dura means tough. That's where we get our words like durable from. So dura mater means tough mother. And the dura mater is divided into 2 layers, and it is the toughest of the 3 layers of meninges. So the outermost layer is called the periosteal cranial dura, and this one is actually fused to the periosteum of the cranial bones that we talked about in chapter 7. So remember, the periosteum is that outermost layer of a bone. So that is actually going to fuse with this outer layer of the dura mater. There is no epidural space, so that would be a space above the dura mater in the cranium. However, in the spinal cord, there is an epidural space, which is discussed in chapter 13. And then the bottommost layer of the dura mater is called the meningeal cranial dura. So this is the innermost layer as shown here. So the meningeal layer can actually extend down into the brain in places, and it creates these structures called dural folds. And there are 3 major dural folds, although we're not going to cover those this semester. And then there is a gap between the 2 layers, and this gap is a sinus. So this is called the dural sinus, and this is a place where blood is drained from the brain as well as CSF. The largest collecting veins are located within the dural folds, and we'll mention those in a bit when we talk about recycling CSF and the blood supply to the brain. There is a small space underneath the innermost layer of the dura mater called the subdural space. So remember, sub means below, so below the dura membrane. Normally there's nothing here, so normally this space is completely empty, except in disease and trauma conditions like with a subdural hemorrhage that we'll talk about when we talk about blood supply. So the middle layer of the meninges is called the arachnoid mater, and it actually resembles a spider web, and that's how it's got its name. Arachnoid means spider in Latin. So that is the middle layer. And the subarachnoid membrane has a space underneath it called the subarachnoid space, and this is the part that actually really looks like the spider web. It's made of a delicate network of collagen and elastic fibers called the arachnoid trabeculae, and this space is going to be filled with the cerebrospinal fluid. So the same CSF that runs through the ventricles also runs through the subarachnoid space. And then the pia mater is the innermost layer. So this is the thinnest and most delicate of all of the layers. And this one is actually anchored to the outer surface of the brain by astrocytes. The pia mater can also surround cerebral blood vessels that go down and penetrate into the brain. And again, remember that the pia mater is anchored to the astrocytes, and the astrocytes are wrapping around these blood vessels to create the blood-brain barrier that we discussed when we talked about the different types of neuroglia in chapter 12. So the meninges function to offer protection from physical forces, so they help prevent excessive movement of the brain within the cranial cavity. So even though we have the hard skull bones that can also help to protect the brain also needs to be kept from hitting up against the skull bones, which can damage it. So the meninges act sort of like seat belts to hold the brain in place to keep it from moving and sloshing around inside the cranial cavity. And then the meninges also circulate and drain cerebrospinal fluid, which we'll talk more about in the next section. In this section, we're going to look at cerebrospinal fluid, or CSF. We're going to look at its functions, how it's created, circulated, and reabsorbed, and then we'll talk about the condition called hydrocephalus. So cerebrospinal fluid has several important functions. One, it does help to cushion the brain against physical trauma. So we already mentioned that you have the cranial bones that can protect the brain. You've also got the meninges that help hold the brain in place within the cranial cavity. And then the fluid of the CSF helps to cushion the brain, so if there is an impact, it helps to absorb some of the shock and lessens the force that's actually placed on the brain itself. It's also a very buoyant fluid, so it supports the brain, and it basically reduces its weight by a lot. So the brain's like 150 grams if it were sitting in the skull cavity with no fluid, but when you add in the CSF, it makes it only like 50 grams. So think of it as like it's floating in the water. This is also like when you're trying to lift something heavy, it's always easier to lift it if it's in water than when it's out of water. So the water provides like a buoyant effect, and CSF does that for the brain. CSF is also very important for circulating nutrients, gases, and waste throughout the brain. Remember that the brain has a blood-brain barrier, so it doesn't have direct contact with the blood. So CSF acts as sort of the circulatory system for the central nervous system. It's going to bring nutrients and oxygen to the cells and carry away carbon dioxide and other waste. So cerebrospinal fluid is produced by a structure called the choroid plexus. And here's a close-up view of a choroid plexus down here in the bottom right corner. So the choroid plexus is made up of specialized ependymal cells. So remember, ependymal cells were those neuroglial cells that line the ventricles within the brain. And then within the part that forms the choroid plexus, the ependymal cells are very close to permeable capillaries. And so the ependymal cells can take fluid from the brain, from the blood. It filters that fluid to produce the CSF, and fluid can also go back the other way from the CSF back into the blood through the choroid plexus as well. But mostly the choroid plexus plays a role in producing the CSF. So it does it by filtering the blood and altering the blood until the liquid component is just what it needs to be for the neurons to do their best jobs. And the CSF will not have blood cells in it, so all of the white blood cells and red blood cells will stay behind. So it's basically just going to be the plasma of the blood with the concentrations of proteins and ions altered just a little bit. So choroid plexus can be found in the third ventricle, shown here, also in the fourth ventricle, shown here, and it also extends up into the lateral ventricles and sits on the floor of both the left and right lateral ventricles. So all four ventricles have some amount of choroid plexus within them that is producing CSF. So the CSF will also circulate through all of the ventricles, the passageways, and then down into the central canal and out into the subarachnoid space, as we'll see on the next slide. So looking at the circulation of CSF, remember CSF is produced by the choroid plexus, which is located in the ventricles. So here's some of the choroid plexus in the lateral ventricle, here's choroid plexus in the third ventricle, and here's choroid plexus in the fourth ventricle. The CSF then flows through the ventricular system and the passageways. We went over this earlier, but just as a quick review. So from the lateral ventricles, it would flow through the interventricular foramina into the third ventricle. Then it would flow through the cerebral aqueduct into the fourth ventricle, and then it would go down into the central canal. It then can pass into the subarachnoid space from two openings in the fourth ventricle, which is shown here in this image. So you can see the red arrows are representing CSF flow in this image. So it's flowing through the ventricles, through the central canal, but it also can come out into the subarachnoid space here. And then it flows throughout the subarachnoid space all around the brain and the spinal cord. So it's completely on the outside of the brain and spinal cord as well as being on the inside. And then it's reabsorbed through structures called subarachnoid granulations that are located in the superior sagittal sinus, and this is how the fluid goes back into the bloodstream. So the superior sagittal sinus is one of those dural sinuses that we mentioned that sits between the two layers of the dura mater. So we had that periosteal layer of the dura mater and the meningeal layer of the dura mater. And between those, we have that dural sinus. This superior sagittal sinus is one of the largest of the dural sinuses, and it's located in the falx cerebri dural fold. And in the superior sagittal sinus, you have these arachnoid granulations, which are extensions of the subarachnoid space up into the dural sinus. And these structures allow the reabsorption of CSF back into the bloodstream. So this is venous blood that you would find in these sagittal sinuses. This comes from veins, and we'll talk about blood supply in just a moment. And this is also where CSF is reabsorbed. So remember, CSF is made from the blood and then it returns to the blood. So we make about 500 milliliters per day of CSF. but our brain and spinal cord and all of those spaces can only hold 150 milliliters total. So if you do the math, right, if we can only have 150 mils total in our brain and spinal cord, but we're making 500 mils a day, that tells you that the entire amount of your CSF is recycled about every 8 hours. So the CSF that you have now is completely different CSF than you had 8 hours ago. So it's constantly being remade and then recycled, then remade, then recycled over and over again to keep it fresh and to make sure the brain has constant access to fresh nutrients and is getting rid of the waste that it needs to get rid of. However, if we have a problem with the reabsorption of CSF, this can cause issues. If we're making 500 mils a day and we only have room for 150 mils, if we can't reabsorb it, this is going to cause issues. And this is a condition called hydrocephalus. This is when you have a buildup of CSF due to blockage. So there could be a blockage somewhere in the pathway that we went over on the previous slide. Or more likely, there's a problem with the reabsorption and the CSF is unable to be reabsorbed back into bloodstream. Just because you can't reabsorb it doesn't mean you stop making it. So you keep making it, but it starts building up because you can't reabsorb it. In children, this can cause an enormous enlargement of their skull. If you remember from chapter 7, children have those fontanelles and their skull bones are not yet fused together. So the fluid just builds up within the head, pushes the skull bones outward, and doesn't actually affect the brain. So because those sutures are not yet ossified, the skull can expand. In adults, it's actually much more serious because you do have those sutures in the skull, so the skull cannot expand. So the fluid then pushes backward and distorts and damages the brain itself. In this section, we'll look at the brain's blood supply, talking about the major arteries and veins, and then some conditions such as hemorrhages and strokes, also called cerebrovascular accidents, and we'll also revisit the blood-brain barrier. So the brain needs an extensive blood supply to meet its oxygen and energy demands. The brain is actually quite a glucose hog. It uses up about 30% of all the glucose that your body takes in or creates. It also needs a lot of oxygen because it has no way to store extra oxygen, so there is no myoglobin in the brain. So it needs a constant oxygen demand. And if you think about all of those sodium-potassium pumps that are always working to reset the balance of sodium and potassium ions after action potentials and because of the leak channels, just think of all of the ATP that the brain needs to keep all of those pumps running. And the mitochondria within the neurons, in order to make all that ATP, needs a constant high supply of oxygen. So your brain is one of the organs in your body that uses up quite a lot of oxygen on a second-by-second basis. So if you interrupt the blood flow to the brain, it can quickly cause unconsciousness, sometimes even just within seconds. And if your brain goes more than 4 minutes without an oxygen supply, i.e. a blood flow, then you can actually have permanent neural damage that can occur. This is why when people are brought back to life using CPR, there's always a concern that they may have some brain damage if they have actually had no blood supply going to their brain for longer than 4 minutes. So the brain, because it needs all of this glucose and oxygen, has quite an extensive arterial supply. So arteries, if you remember, carry blood away from the heart and towards the body. So arteries are carrying the oxygen and the fresh nutrients. So if you look at the base of the brain, you can see that there are a number of arteries that go up into the brain and then branch extensively. There's this whole network that sits right underneath the base of the brain, a bunch of different arteries. I want you to know the two major arteries, which are the internal carotid arteries. There's one on the left and the right. And then you've also got two vertebral arteries, again, one on the left and the right. So the vertebral artery and the internal carotid arteries are your major arteries that bring the largest supply of blood up to the brain. But again, you've got a lot of redundancy and a lot of cross connections in an attempt to try to keep the brain from running out of oxygen and glucose. And then veins drain blood. So they take, there is oxygen poor blood. It takes the blood that has the oxygen already removed from it. It's usually full of waste products, has no nutrients left. And the veins are draining into the dural sinuses. Remember, the dural sinus again is that space between the two layers of the duramater. And we've already talked about the largest of these dural sinuses. It's called the superior sagittal sinus. There are other sinuses, but the veins drain into these sinuses, and then all of the sinuses drain into the internal jugular vein. So the internal jugular vein is the major vein that drains blood from the brain to return it back to the heart. So if you have a severe head injury, this can damage blood vessels in either the meninges or the bones, and this can cause bleeding within the skull, and this can be very dangerous. So the first type of issue with bleeding is called an epidural hemorrhage. So an epidural hemorrhage occurs when damaged blood vessels are forcing blood between the duramater and the cranium. So remember, the outer layer of the duramater is normally tied to the periosteum of the cranial bones, so it takes quite a high pressure bleed to force blood into this area. And in this picture here, the dark pink line represents the duramater. So on the left here, we have an epidural hemorrhage, which is going to be between the duramater and the skull. And these types of hemorrhages are usually caused by ruptured arteries within broken cranial bones. And so the blood quickly can fill up in this space. In contrast, a subdural hemorrhage is when blood pools between the duramater and the arachnoid mater, and that's shown on the right side. So our duramater is still attached to the cranial bones, but the blood is building up underneath the duramater, so between the duramater and the arachnoid mater. Now you can notice that in both cases, neither situation is ideal, because not only are you bleeding and losing blood, but the pressure of the blood pooling in the cranial cavity is forcing the brain to be squished down. So this can eventually lead to damage of the brain tissue itself in both cases. Subdural hemorrhages are more common than epidural hemorrhages, again, because normally that dura is attached to the skull, so it's kind of hard to force the blood in between the duramater and the skull. And the subdural hemorrhages usually happen when you've got damage to those venous sinuses, like the superior sagittal sinus that we were just talking about. So the severity of either types of these hemorrhages depends on whether it was an artery or a vein that was damaged and starts bleeding. Arteries have higher pressure, and because they have higher pressure, the blood can pool more rapidly. So if you think about all those gross movies you've probably seen, when you see the blood shooting across the room, that's coming from an artery. A slow drip of blood is coming from a vein. So arteries have much, much higher pressure. So if you damage an artery, such as shown here on the left side of the picture, that blood will pool up very rapidly and cause effects very rapidly. In contrast with a venous damage within the skull, sometimes effects may not even become apparent until days later. This is why if you damage your head, they want to do a CT scan of your head, even if you feel fine, because you could have a subdural hematoma caused by a vein that broke that you have no symptoms whatsoever for, because the veins leak very slowly, and it can take hours, days for that blood to build up to cause any noticeable effects. So epidural hemorrhages are usually arteries, and these are rapidly fatal. So an untreated epidural hemorrhage is 100% fatal, actually. Subdural hemorrhages are usually veins, so these are slower. They can develop over time, and these are dangerous just because people don't know they have them. They hit their head, they think they're fine, they go on about their day, and then it's not until a couple of days later they might start displaying symptoms, and then they have to be able, them or their doctors have to be able to connect the later symptoms with the head injury that might have happened a few days earlier. So subdural hemorrhages can develop more slowly, but that can cause its own set of problems. Another type of blood supply issue is called a cerebrovascular accident, or CVA. This is what people more commonly call a stroke. And this occurs when the blood supply to a particular region is cut off. And this is usually because of a blood clot. So in this picture here, if I have a blood clot in this artery, then all of the area that's normally supplied by the artery can no longer get blood, can no longer get oxygen, and within 4 minutes you're going to start getting neuron death. And this is what it would look like on a scan, an area of dead neural tissue within the brain. And so the effect of a stroke depends on which region was affected. So again, you've got lots of blood vessels within your brain, so depending on which one the clot got stuck in is going to determine the outcome. And we'll talk a little bit in the in-class activity about some of the types of effects you can get from different types of strokes. So let's revisit the blood-brain barrier. So if you remember from chapter 12 when we talked about astrocytes, the blood-brain barrier is created by astrocytes that wrap around blood vessels. So in this image up here, the little red circle in the middle is a blood capillary, and these large purple structures all the way around it are the feet of astrocytes. So astrocytes completely encircle these capillaries. They have tight junctions between one foot and another, so that nothing is passing from this blood vessel out to the neurons if the astrocytes do not let it. So anything that will damage astrocytes will damage the blood-brain barrier as well. So there are a few special cases within the brain where you don't have the astrocyte blood-brain barrier, but there still is a way to protect the neurons from the general contents of your blood. And for example, there is a blood CSF barrier in places where you have the choroid plexus. Instead of having astrocytes that are controlling what comes in and out of the brain, you've got those ependymal cells that are doing the same job. So the blood-brain barrier allows the interstitial fluid that surrounds the neurons within the brain to be tightly controlled. And this is to promote the normal function of the nervous system. So for example, a lot of those neurotransmitters that we talked about in chapter 12, like norepinephrine and even serotonin and dopamine, they also work as hormones in the blood. And if they're in the blood, you don't want them to get anywhere near the neurons, because then they would act as a neurotransmitter and they might start sending signals that you didn't intend for them to send. So for example, the hormones that are in the blood can't cross the blood-brain barrier, so that they can't get into the brain and act as inadvertent neurotransmitters. So this is just one example of why you don't want the general contents of the blood to be in contact with the brain. And this affects the medical field because a lot of prescription medicines cannot cross the blood-brain barrier. So you take something orally, it goes into your stomach, it's absorbed into your bloodstream, and then unless it's made in a specific way, it cannot get into your brain. It can treat other parts of your body, but it cannot cross the blood-brain barrier. So there's a lot of pharmaceutical research and money that's poured into developing drugs for neural conditions that can cross the blood-brain barrier. So if you have any type of neural or mental condition that you take medication for, just know that there was a lot of research that went into whether or not that drug could actually get to the brain where it's needed. And if a particular drug cannot cross the blood-brain barrier and it's needed to treat a neural problem, it would have to be injected straight into the CSF using a lumbar puncture. And we'll talk more about lumbar punctures in chapter 13 when we talk about the spinal cord. But the lumbar puncture is the most common way to either sample CSF, so take it out for study, or to put something into CSF because you need something to go into the brain that would otherwise not cross the blood-brain barrier. In this section, we're going to look at the structures of the brainstem, which include the medulla oblongata, the pons, and the midbrain. So we'll start with the medulla oblongata, which is the most inferior brain region. So this is where the brain is actually connecting up with the spinal cord. And the medulla oblongata contains 3 major groups of nuclei. And so a quick aside here on terminology that we use when talking about the CNS, so the brain or the spinal cord. When you hear the word nuclei in reference to the organ of the brain or spinal cord, this means that it's a group of neuronal cell bodies that have the same function. So this is different from the nucleus of a cell. So don't let that confuse you. So in the brain and spinal cord, a nucleus or nuclei are collections of neuronal cell bodies that are working together to produce the same function. Also, when you see the word tracts or fibers, these are groups of axons that have the same destination, so they are called tracts. So the functions of the medulla oblongata go along with its three major groups of nuclei. So its first major function is that it controls visceral functions. This is going to include coordination of your different types of autonomic reflexes. So for example, it can help adjust your heart rate, either speed it up or slow it down. It can adjust your breathing rate as well. It also can control other reflexes like sneezing, coughing, and vomiting. And we'll talk more about different types of reflexes and even how the medulla oblongata helps coordinate these in later chapters. The medulla oblongata also contains the sensory and motor nuclei of 5 of the cranial nerves, so nerves 8 through 12, so 8, 9, 10, 11, 12. And again, nuclei are groups of cell bodies that are doing the same function. And the third group of nuclei in the medulla oblongata act as relay stations for tracts going back and forth between the brain and the spinal cord. And again, when we say tracts, we're talking about a group of axons with the same destination. So in this particular picture, you can see the relay stations. It's got different arrows pointing to them. I'm not going to go over the different names of the relay stations, but you can also see the autonomic centers here as well, and I am going to talk about a couple of these in more detail. So important areas to know for the medulla oblongata. The first one I want you to know is the reticular formation, which is shown in this image as this long cylindrical oval-shaped yellow structure. And the reticular formation plays a key role in maintaining our alertness and our consciousness. And in fact, if you damage this structure, it can result in an irreversible coma. In other words, you become permanently unconscious, so you can't be awake, you can't be conscious, you can't be alert, because the reticular formation's job is to keep you alert, awake, and conscious. The reticular formation is also very important for your habituation to certain stimuli when you're sleeping. So it learns what to wake you up for and what to let you sleep through. So for example, if you live next to train tracks, when you first moved in, you might have woken up every time a train went by, but eventually the reticular formation learns that that's a stimulus you can ignore, and so you start being able to sleep through it and it doesn't wake you up anymore. So when you habituate to certain sounds, that is your reticular formation. This is also why if you sleep in a new bed in a new house or a new apartment for the first time, every little noise can wake you up. That's your reticular formation that hasn't yet habituated to the sensory information in that new environment. The reticular formation also helps control some of the autonomic functions. So there are other specific reflex centers that we're going to mention in a minute, but the reticular formation can also help with these autonomic functions. It is also part of the pain gateway, whereby pain signals get to the brain. And so some of the way that the brain can anesthetize or cause you to ignore the pain signals also goes through the reticular formation. And the reticular formation also has some areas that help with the somatic motor pathways, in particular the ones that help you maintain your balance and posture. Another two key areas of the medulla oblongata are the reflex centers, which are shown here as the red blob and the purple blob. So you've got both a cardiovascular center and a respiratory rhythmicity center. And we're going to go into more detail in these next semester when we get to the cardiovascular and respiratory systems. So the next structure of the brainstem is the pons, and the pons is shown in blue. It sits right above or superior to the medulla oblongata. And the pons, it plays a key role in linking the cerebellum to other regions of the brain. So if you look at the lateral views shown here, the pons is where the cerebellum, or that small little brain in the back, is actually connected. And so the pons, a lot of its role is to actually relay signals back and forth from the cerebellum to the spinal cord, from the cerebellum to the cerebrum, etc. So it has four main components, so four main functions. One is that it has the sensory and motor nuclei of four of the cranial nerves, so cranial nerves 5 through 8, so 5, 6, 7, and 8. 8 actually has some of its nuclei in both the medulla oblongata and the pons, that's why it's listed twice. The pons also controls respiration, so it doesn't initiate it. So the medulla oblongata has the centers that initiate respiration, and then the pons controls those centers to help adjust those rates of respiration. The pons also has relay centers for messages sent to and from the cerebellum. As I just mentioned, it's a big relay for stuff going back and forth from the cerebellum. And it also has special fibers that allow communication between the left and right sides of the cerebellum. So we'll talk a little bit later about specific fibers that connect the left and right hemispheres of your cerebrum, but the cerebellum also has two hemispheres that need to talk to each other, and they do that through fibers that pass through the pons. So two of the centers in the pons, I'm just going to mention them now and then you're going to learn more next semester. The apnoistic and pneumotaxic centers are these two centers inside the pons. They're shown in purple in this image. These adjust the activity of the respiratory nuclei in the medulla oblongata. And again, we're going to go into more detail in next semester when we talk about the respiratory system. Also notice that the reticular formation is still running through the pons, although it started down in the medulla oblongata. So you can still see it, this long, thin, yellow structure that's running up and down throughout the brainstem. So it started in the medulla oblongata, but it's continuing through the pons. And then the last part of the brainstem is the midbrain, and the midbrain is colored in this image in green. So on the anterior view, you can only see a little bit of the midbrain shown here with the two green up and down columns. Then if you flip it over and look underneath, you can see a lot more of the midbrain. Again, it's showing in the green section here. So the first part that we are going to talk about is the part where you see these they look like four little balls. So two on top of each other over here and two next to each other over here. These are called the tectum, and they're also called the corpora quadrigemina, but I'll tell you that in neuroscience circles, they're just referred to as the tectum, which is a little easier to remember. So these contain two different structures. So the little balls on the top on both sides are called the superior colliculi, and again, if you just talk about one, it would be a superior colliculus. These are going to receive visual input and control your visual reflexes. So for example, your response to bright light, also your response to like something flying past your field of vision, your eyes track it automatically, that's your superior colliculus. Then the two little balls underneath, these are the inferior colliculi, and again, a singular one would be a colliculus. These receive auditory input and control your auditory reflexes. So like if you hear a loud sound and your head whips toward the sound before you can even think about it, that is your inferior colliculus, and that is an auditory reflex. So I like to tell students to remember which one is which. Remember that most of us depend more on our vision than on our hearing. So we tend to be very visual creatures. We like to see things. So think about vision being superior to your auditory. So the superior colliculus is vision, the inferior colliculus is auditory. Now notice these are not your main visual and auditory centers. Those are up in the cerebrum. These are for visual and auditory reflexes only. So at the midbrain level, we're only talking about reflexes. Another major structure, so these two green columns that you could see on the anterior view, these are bundles of fibers that are descending from the cerebrum down to the cerebellum and the spinal cord. So these are just bundles of axons. So again, a little bit of neuroterminology. When you see bundles of fibers, that's the same thing as saying a bundle of axons. The midbrain also contains the motor nuclei for two of the cranial nerves. So cranial nerves number 3 and 4. The first two cranial nerves are actually part of two of your sense organs. Cranial nerve 1 is part of your eyes, and cranial nerve 2 is part of your nose. A couple of other areas in the midbrain. So if I cut across the midbrain here, as shown in this cross section at the top right, there is an area called the substantia nigra. This actually looks black, and nigra in Latin actually means black. So this is the area with the black substance. That's how it got named. This contains dopaminergic neurons, which regulate the basal nuclei, and we're going to learn more about the basal nuclei later, but they actually help to control subconsciously your muscle tone and learn movements. And when you have Parkinson's disease, the neurons that are found here in the substantia nigra are usually the first ones to die. And when I say a dopaminergic neuron, that means it's a neuron that uses dopamine as its neurotransmitter. There's also an area in the midbrain called the red nucleus, shown right here. It's got a red color because of the proliferation of blood vessels in this area. This area provides subconscious control of your upper limbs, as well as the muscle tone you have when you're not consciously thinking about contracting your muscles. And then if you notice, the reticular formation that we mentioned before also comes all the way up into the midbrain. And in the midbrain, the reticular formation, there's a specific area of it called the reticular activating system, which is abbreviated RAS. And this is a specialized part of the reticular formation, and this is the part that plays that key role in maintaining consciousness. So it's this part in particular that if it's damaged, you'll be in an irreversible coma. In this section, we're going to talk about the cerebellum, and we'll also talk about ataxia, which can result from damage to the cerebellum. So the cerebellum has two primary functions. The first one is that it adjusts your postural muscles to maintain your balance and equilibrium. So the reason that you can stand up straight or sit up straight in a chair without thinking about it is due to your cerebellum. It also programs and fine-tunes learned movements. So when you've learned to do a particular movement that you do over and over again, you no longer have to think about what you're doing, and that is because the cerebellum has taken over and now controls and fine-tunes that movement. And so a lot of times you'll hear people refer to muscle memory, and your muscles don't actually have a memory. The memory of your movements actually is stored in the cerebellum. So when you learn, for example, how to ride a bike or play a piano or do certain moves in sports or a type of dance, when you first start, you can be very clumsy, very clunky in your movements. But as you repeat those movements over and over again, the cerebellum learns those movements, and it learns the precise nature of moving those muscles until you can do it without thinking. And so the cerebellum is basically all about the fine control of your muscles to produce smooth movements. So the commands to move your muscles, so when you decide I'm going to raise my hand, for example, that comes from the cerebrum, as we'll talk about later. There's an area of the cerebrum called the motor cortex. But the cerebellum does the fine-tuning of these movements, and it also stores the memory of these specific types of movements that you do over and over again, so like repetitive movement tasks that you've learned. Some of the cells in the cerebellum receive input from up to 200,000 synapses on their dendrites and soma. And so these are some of the neurons in the brain that receive the most information on their receiving ends. And here's what one of the neurons look like. So this is a cerebellar Purkinje neuron. And what you're seeing in this picture, these are all dendrites. You can see a little bit of the axon down here at the bottom, but look at the number of dendrites. So think about how many signals this cell can be receiving from other neurons. And so you got a heck of a lot of summation going on in these neurons. So what kind of information are these neurons getting? Well, they actually get a copy of the motor command. So as I mentioned before, the motor command, when you decide to make a movement, that starts up in the cerebrum, and the cerebellum gets a copy of it. And the cerebellum is also going to get what we call proprioceptive info, which is sensory info on where your joints and bones and muscles are in relative to space. So like this is how you know, for example, you can close your eyes and know that you have your arm in the air, like raised above your head. This is like the sense of your body position. So the cerebellum gets a copy of this so it can know that your body is doing what it's expecting it to with that motor command. It also receives visual information, which can help with like balance, so you can orient yourself against the horizon. And it's basically taking all of this sensory information. It also gets touch, balance, and auditory signals. And it's taking all the sensory information and basically saying, are we doing what we want to do with this motor command? Because remember, it gets a copy of the motor command. So the motor command is like, I want to raise my arm. The cerebellum gets a copy of that command as well as a bunch of sensory data. And so the cerebellum is the one that is calculating, okay, are we raising our arm like we wanted to? Is the arm in the position that it's supposed to be? And so it does through trial and error. It learns the precise way to move the muscles to accomplish the movement that you wanted to do originally. And really, watching mammals learn how to walk is a great way to see the cerebellum in action and to see the cerebellum learning how to coordinate those motor commands with the sensory input that it's getting. So we're going to take a quick little break here, and I'm going to show you a couple of videos. So when you watch animals learn how to walk, including humans, you can see the cerebellum in action. So it's taking the motor command, which is the moving of the legs, you want to stand, that's what your goal is. And it's comparing the sensory information, right? So he just lost his balance, right? So it's comparing balance information from the position of your joints, visual information. It's taking all of that information and it's comparing it to the motor command, what we actually wanted to accomplish. And at first, there's a lot of dissension between what we want to do and what we actually do, but this is the cerebellum actually learning to fine-tune these movements. So in all of these cases, you're actually watching the cerebellum undergo a learning process. And then of course, once the cerebellum has made this learning process, now your movements are smoother, no jerkiness, no imbalance. Now the cerebellum has learned. And just one more cute video of a human learning to walk, because this one is really cute. Whoa! Still got a way to go, but he's learning. And in terms of the different parts of the cerebellum, I just want you to know that the arbor vitae is the name given to the branching white matter within the cerebellum. So early anatomists thought it looked like a tree, and they knew that it was important for life. So arbor vitae in Latin literally means tree of life. Vitae means life, arbor means tree. So that's the name given to the white matter in the cerebellum. And again, remember, white matter is myelinated axons. So these are axons carrying information from one place to another. So ataxia is a term that's used to describe any disturbance in motor, aka muscular coordination. So it's quite a broad term, so any problem with movement is called ataxia, but one of the most common types of ataxia is caused by cerebellar damage, and this could be either through trauma like a head injury or a stroke. There is also some forms of genetic ataxia that you can be born with. Now, ataxia can also be caused by alcohol intake. However, it's temporary and it only lasts while you're under the influence. And this is why you see people who are drunk essentially have problems with their muscle coordination. It's because the alcohol is inhibiting the cerebellum, and this is why they stumble and can't walk straight. And this is why the cops have that, walk on a straight line test. So physicians often use what is called the finger to nose test to check for cerebellar damage, and this is when they ask you to place your finger on their finger and then bring it to your nose and then back again, and they're looking for any shakiness in this movement. It should be a very smooth movement, so they're looking for any shakiness or impreciseness, like not hitting your nose, and that tells them that there could be something potentially wrong with your cerebellum. And as I mentioned, cerebellar ataxia is one of the most common types of ataxia, but the specific results can depend on which part of the cerebellum is damaged. So damage to one area may only affect visually guided movements, damage to another area may only affect goal-directed movements, and damage to a third area may only affect your balance and your posture. So I have another video I'm going to show you. This is what cerebellar ataxia looks like. So we're going to look at a gentleman who has cerebellar ataxia, and so this will show you exactly what this looks like. And so we're looking at the finger to nose test that I mentioned, and this shows the inability to control the hand. So this is his good hand, so only one side of his cerebellum is damaged. So with his good hand, it's just like you guys should be able to do. Bring your finger straight to your nose and take it away, and it should be a nice smooth movement. And then if you take a look at his affected hand, you can see the difference. All right, so notice the jerky movements, it's not smooth. This is because the cerebellum on that particular side is unable to do that fine tuning. You can still move. And then notice how it learns. Notice the more he does it, the better it gets. Although when he stops and starts again, it's no longer learned because of the damage. So it can do short-term learning in this case, but it can't do long-term learning. So again, this is the good hand. This is what everyone should be able to do. And again, you notice the jerky movements. You can still move, it's just not smooth. And the more he does it, though, it starts getting a little bit easier and a little bit easier because it's doing some short-term learning. And again, just showing how he doesn't have the smooth movement. And so if you want to know, in this case, this was a case of where this gentleman was riding a motorcycle without a helmet. And so his damage to his cerebellum came about because of an accident, and he wasn't wearing a helmet to protect his head. So in this section, we're going to look at the Diencephalon, which includes the pineal gland, the thalamus, and the hypothalamus. So we'll start with the pineal gland, but first let me orient you to this view. So in this view, we've already talked about the medulla oblongata, which is in this salmon color, the Pons, which is in the blue color, and the midbrain, which is in the green color. And so now we're looking at the Diencephalon, which is in this mauvey purpley color here at the top. So the pineal gland is this little small gland that sits right above the superior colliculi and right between the two thalami that we're going to talk about in a moment. So the function of the pineal gland is to produce melatonin. It used to be called the pineal body, so you may still see that in some older textbooks, but then once they realized that it produces a hormone, it got renamed to a gland. And melatonin is a hormone that's most known for its role in day-night cycles. So light that comes in through the eye and then passes through a region of the hypothalamus called the suprachiasmatic nucleus that we'll talk about in a few slides. And then the light information is passed on to the pineal gland. And with light exposure, there is inhibition of melatonin production. And then in the dark, there is stimulation of melatonin production. However, we've learned a lot in recent years about how melatonin doesn't just help you get a good night's sleep, but it's also important for good health in many areas of the body. So this image here just shows you a few. For example, melatonin can help accelerate wound closure in wounds that are in your skin. It can help promote good things in the kidney, in the bladder. It can help promote bone growth. It can help promote axonal regrowth and Schwann cell regeneration if you have peripheral nerve damage. It can maintain several areas of the CNS. Again, it can reduce stress in the liver. It can also reduce atrophy in muscles. So basically, melatonin is really good for maintaining a lot of the different systems of your body, but you only produce it in the dark and you only produce it at night. So there's also been a lot of research about how now that we have electricity and we're exposing ourselves to light at night, how this is actually a bad thing. So exposure to room light in the evening hours actually will suppress your melatonin onset, and then when you do make it, it will actually shorten the duration that you make it. And because melatonin does such wonderful things to help repair and maintain your body, having this suppression of melatonin because of exposure to light at night is actually causing all sorts of health problems. And here's a conclusion from this article. These findings indicate that room light exerts a profound suppressive effect on melatonin levels and shortens the body's internal representation of night duration. Hence, chronically exposing oneself to electrical lighting in the late evening disrupts melatonin signaling and could therefore potentially impact sleep, thermoregulation, blood pressure, and glucose homeostasis. So, for example, lack of melatonin is now being linked to things like obesity, diabetes, and cardiovascular disease. I'm going to post an article from Harvard Health on D2L if you want to read up more about the effects of light exposure at night and the detriments on your health. So the next major structure in the diencephalon is the thalamus. The thalamus is the gate or the relay point for all ascending sensory information other than olfactory information. So any other sensory information, vision, hearing, taste, smell, touch, the proprioception that we mentioned earlier, all of that is going to come through the thalamus. And so the thalamus actually will filter this information and only pass some of it along. So it helps you to focus by allowing you to ignore all of the sensory input that is constantly bombarding you. So if you think about it, right, after you put your clothes on, maybe when you first put your socks on, you recognize or feel the texture of the sock against your foot, but you're not consciously aware of that all day long, right? At some point, you're no longer thinking about the feel of the sock on your foot. So the thalamus is basically relaying just specific information that you need to focus on whatever task that you're doing, and all of the other extraneous sensory information gets filtered out. So people who have a hard time focusing might have a deficit with their thalamus's ability to filter out the unwanted sensory information. So there are multiple thalamic nuclei, and again, remember, a nuclei is a collection of neuronal cell bodies that do a similar job, and they are color-coded in this image from your textbook. This is just showing one of the two thalami, and you can see that there are multiple different nuclei, and they're color-coded to where they go in the brain. So there's a lot of areas for sensory input like auditory and visual and general sensory input. And then you've also have some nuclei that play a role with the basal nuclei that we're going to talk about soon, and the limbic system, which we're going to talk about soon. You don't need to know the different nuclei in the thalamus, even though they're listed in your textbook. Next semester in AMP2, we will mention the medial geniculate body and the lateral geniculate body when we talk about vision and hearing, but that's next semester. And then here is another image just to help you see how the thalamus works as like a relay system. So I've got my thalamus here with neurons inside of it, and it's getting information from my sensory systems out of my periphery, and it decides what to send on to the cortex, which is, remember, the cerebral cortex are the cells, the neuronal cell bodies that are in the outer layer of the cerebrum. So it decides what information to pass upward for you to be conscious about. So as I mentioned, there is a left and right thalamus. A lot of times textbooks will only show you one, so you kind of forget that there's actually two of them. So you do have one on the left side and the right side, and each one of the thalami has all of these different nuclei in it. And then they are connected to each other by a little piece of gray matter called an interthalamic adhesion. And you'll also often see this in the models that we have in lab. And so if you see this little dot here in the middle, and I'll point an arrow to it, that is the interthalamic adhesion that would be connecting the left and the right thalamus. And also when you're looking at pictures of the ventricles, remember that third ventricle that's right in the middle of the brain, that hole that you see there in the third ventricle is actually where that interthalamic adhesion passes through. So that's where the two thalami are connected to each other. And the last structure of the diencephalon is the hypothalamus. So if you look at the picture down here, here's our thalamus up here. Here was the interthalamic adhesion that connects one to the other. The hypothalamus sits right under the thalamus. So the name hypo, remember hypo means below or lower. So hypothalamus, it's below the thalamus. And the hypothalamus has a lot of nuclei as well. Again, these are collections of cell bodies that perform the same function. However, I do want you to know several of these. So the hypothalamus has the mammillary bodies, which are shown here in this sort of dark pink color. These play a role in sensory input and the motor control of eating, like licking and swallowing and chewing, as well as a small role in memory. You also have autonomic centers, which are shown here in the orange and blue. These control areas that regulate heart rate and blood pressure. Basically, these are the control centers for the sympathetic and parasympathetic divisions of the nervous system. So we're going to talk more about these guys in chapter 16. You also have the paraventricular nucleus, which is shown in this small green one up here at the top. The paraventricular nucleus secretes the hormone oxytocin, which we'll talk more about in A&P2. You also have the preoptic area, which is shown in the yellow rectangles here. This area, your body talks about it, how it regulates body temperature, but it also plays a role in sleep regulation. This is where you have the switch that switches your brain from the sleep state to the wake state and back again. Then there's also the suprachiasmatic nucleus shown in green at the bottom here, and we mentioned this one a little bit earlier. This one regulates circadian rhythms, so it gets light input from your eyes, and so it takes that light and helps to regulate the circadian rhythms within the body. Then there's the supraoptic nucleus, which is the front side of this little green guy down here at the bottom. And the supraoptic nucleus secretes another hormone. This one is antidiuretic hormone, also known as ADH. And again, you'll get more information on ADH in A&P2. So the pituitary gland is also attached to the hypothalamus through a stalk called the infundibulum. And you can see that the axons of some of the hypothalamic cell bodies actually come down through the infundibulum so that they can release their hormones out of the posterior pituitary. And we'll talk more about the pituitary gland and the secretion of all of its hormones in A&P2. So the hypothalamus has a lot of functions, which makes sense because it has a lot of different nuclei. And so you do need to know all of the functions of the hypothalamus. So one of his functions is the subconscious control of skeletal muscles. So remember, skeletal muscles are normally also called voluntary muscles, but you can have movements in them that are involuntary, and the hypothalamus is responsible for those. So behaviors associated with rage, pleasure, pain, and sexual arousal. So movements that you make when you're in these hyper emotional states, you're not really consciously thinking about making those movements, and that is the hypothalamus that has taken over control of those skeletal muscles. The hypothalamus also controls autonomic functions, things like heart rate, blood pressure, respiration, and digestion. And so it coordinates areas in the pons and the medulla oblongata. And again, we'll talk more about this in chapter 16. The hypothalamus also coordinates activities of both the nervous and the endocrine systems. It is sometimes considered to be the master gland that controls a lot of the secretions within the endocrine system. So it kind of has its legs in two systems. It can be considered part of both the nervous system and the endocrine system. And it also produces a lot of regulatory hormones that control the pituitary gland. And again, we'll talk more about that in AMP2. The hypothalamus also secretes two hormones itself. So it secretes antidiuretic hormone and oxytocin. The hypothalamus also produces emotions and behavioral drives, and we like to tell students to think of these as the 4 F's. So feeding, fighting, fleeing, and I'm sure you can think of an F word that represents reproduction. So your basic behavioral drives that allow you to survive and pass on your genes, those behavioral drives are regulated by the hypothalamus. The hypothalamus also coordinates between your voluntary and your autonomic functions. So for example, if you think about something scary and it kicks in your fight and flight response, the connection there between your conscious thought and the autonomic response takes place in the hypothalamus. The hypothalamus also regulates the body temperature. If you remember from chapter 1, when we introduced you to homeostasis and the homeostatic mechanism, we talked about how the hypothalamus was the control center for the body temperature regulation. So that's where the set point is. So the hypothalamus is comparing input that's coming in from the periphery and deciding if you're too hot or too cold, and then sending signals back out to make changes. And then the hypothalamus also controls your circadian rhythms. So this is both your day and night rhythms. And then in some mammals, it also plays a role in their seasonal rhythms, like animals that hibernate, for example. In this section, we'll look at the limbic system, which is not necessarily a structure so much as it's a functional grouping of structures that also belong to other areas. So as just mentioned, the limbic system is more of a functional grouping of different structures that play a role in motivation, emotion, and memory. So some of the larger structures we've already talked about, like for example, the hypothalamus, part of the hypothalamus, specifically the mammillary body, plays a role in the limbic system, as does a portion of the thalamus, the anterior thalamic nuclei. And we've also mentioned the cerebrum, which is the wrinkled portion on top, and a part of the cerebrum called the cingulate cortex, which is this gyrus right here, also plays a role. So like I said, this is more of a functional grouping of structures that play a role in motivation, emotion, and memory. So the functions of the limbic system are to establish your emotional states. So emotions come from the limbic system. The limbic system also links the intellectual functions of the cortex with the unconscious autonomic functions. This is your motivation side of things. So for example, if you unconsciously sense that you're hungry, this will be passed up to your intellectual area of your brain, and you're going to want to go find something to eat. So the limbic system is what makes you motivated or want to eat when you're hungry. It also facilitates memory storage and retrieval. So these are episodic memories, which are memories of like events, factual memories like what you're learning right now in your AMP course, and emotional memories. We already talked about how motor memories or muscle memory is stored in the cerebellum, but your other major types of memory are handled by the limbic system. So the major structures to know from the limbic system are the hippocampus, which is shown in this dark pink color down here. It wraps around on both the left and right sides of the brain. And the hippocampus plays an important role in learning, and this is also the structure that's responsible for storage and retrieval of your long-term memories. The amygdala, which is shown right here in this dark purple area, the amygdala plays a role in aggression and fear and anxiety. So kind of like what we think of as the negative emotions, although it can play a role in positive emotions as well. The amygdala also plays a role in linking emotions with memories. And the amygdala is one area of the brain that does have major differences in males versus females. So males typically have a larger amygdala, and it has a slightly different structure than the female amygdala. This is thought to be one of the reasons why males typically show aggression more than females. And interestingly, in homosexuals, the amygdala shows a difference as well. Specifically, homosexual males have an amygdala that resembles more of a female amygdala, and homosexual females have an amygdala that looks more like a male amygdala. The amygdala is also linked to PTSD, post-traumatic stress disorder. So if we've known that two people experience a traumatic event, not both of them are going to go on and develop PTSD, but people who have certain structure and size to their amygdala are more likely to develop PTSD than people who don't. So the differences between whether someone develops PTSD or doesn't depends on their amygdala. And then the cingulate gyrus is this gyrus, which is this fold of the cortex that sits right above the structure called the corpus callosum, and it's shown in green here at the top. The cingulate gyrus links behavioral outcomes to emotion. So for example, you decide to do something and the result was either a very positive result or a very negative result. The cingulate gyrus helps you link that corresponding emotion with what your behavior was. So it's kind of like how you learn that certain events have consequences. And of course, the stronger the emotion that's connected to the outcome, the stronger the learning and the connection. The cingulate gyrus is also sometimes called the cingulate cortex, so you may see it referred to that as well. And it seems to play a role in schizophrenia. So when they look at brain scans of people that have schizophrenia versus people who don't, there seems to be a lot of differences in the cingulate gyrus between someone who doesn't have schizophrenia and someone who does. And the last little thing to point out is the fornix. The fornix is shown in this picture in yellow. And the fornix is a tract, which means it's a bundle of axons. And so these are axons that are connecting neurons in the hippocampus with neurons in the hypothalamus. In this section, we're going to start talking about the cerebrum, and we're going to start with its overall organization, and we're going to look at the major lobes, gyri and sulci, and then we'll go into details in the subsequent sections. So as mentioned in the beginning, the cerebrum is the largest region. It's that large wrinkly portion that most people think of when they think of the brain. The cerebrum is responsible for all conscious thought and intellectual functions. So we sometimes call these the higher order functions. And as mentioned in the beginning, the cerebral cortex is the outer layer of neuronal cell bodies that are also called cortical neurons. Cortical because they're part of the cortex, and this is making up that outer layer of gray matter. And the number of wrinkles increases the surface area, and this allows for an increased number of neurons, which provides more processing power. So if you go back to the beginning of the lecture, you can see the cerebral cortex of several different mammals that I compared for you. So terminology, the longitudinal fissure is this line that separates the two hemispheres. So the left cerebral hemisphere from the right cerebral hemisphere. And each hemisphere is divided into 4 lobes: the frontal lobe, parietal lobe, occipital lobe, and temporal lobe. We'll look at those in more detail in a moment. And then if you separate the frontal lobe and the temporal lobe, underneath there is another area called the insula. So let's look at the different lobes. So here's a picture from your textbook where the lobes are color-coded. And then at the bottom we can see the mid-sagittal section. So you can see how the different lobes continue down inside the brain, with the exception of the cingulate gyrus, which is uncolored. And the cingulate gyrus is part of the limbic system, as we discussed previously. So there's the frontal lobe, which is right at the front of the brain and is shown here, and it's in pink in your diagram from your textbook. The frontal lobe has one major wrinkle that you should know the name of, and that is the precentral gyrus, which is shown here in darker pink. This is where you have the location of the primary motor cortex that we'll talk more about in a moment. Other areas that we're going to talk about, which I'm only introducing here, are that you can also find the premotor cortex, the prefrontal cortex, and Broca's area, which is responsible for speech. All of these are in the frontal lobe. Then you have the central sulcus, which is a deep groove that separates the frontal from the parietal lobe. So you can see the central sulcus running right down here. It's the groove. Remember, sulci are the grooves between the gyri. And then in purple, we have the parietal lobe, also shown here in this little GIF animation. And the other gyrus you need to know is the post-central gyrus, which is shown in dark purple. So now that you know that that line that separates the frontal lobe and the parietal lobe is called the central sulcus, the names of the gyri should make more sense to you. So the precentral gyrus is the one in front of the central sulcus, so precentral. And the post-central gyrus is the one behind the central sulcus, or post-central. The post-central gyrus contains the primary sensory cortex, and the other areas that we'll learn about in the parietal lobe are the language processing areas and somatosensory association areas. Then we have the parieto-occipital sulcus. This is back here separating the parietal and occipital lobes. Then you have your occipital lobe, which is in the very back of the brain. The occipital lobe contains the primary visual cortex and the visual association area. You also have a lateral sulcus, which is shown here. My mouse is going over it now. The lateral sulcus separates the parietal and frontal lobes from the temporal lobe on the side, which is shown in this light yellow color. And then finally we have the temporal lobe, which is on the side of the brains. And the temporal lobe has the primary auditory cortex, the auditory association area, and the olfactory cortex. And now we're going to go into a little bit more detail about the structures you can find in the cerebrum. So now we're going to talk about the basal nuclei of the cerebrum and Parkinson's disease. So the basal nuclei are masses of gray matter in the cerebrum that lie deep underneath the lateral ventricles. So you can see in this top picture, these basal nuclei are colored in this dark pink color, and then down here at the bottom with the cross section, you can see them again. So remember, the gray matter that lies on the outside of the cerebrum is called the cerebral cortex, or cortical neurons. And so this is additional gray matter that is deep inside of the cerebrum. Most of the rest of the cerebrum is made up of white matter. So the three major structures that make up the basal nuclei are the caudate nucleus and the putamen and globus pallidus. And these are the three different pink areas shown in this picture. So the basal nuclei function in the subconscious control of skeletal muscle tone and the coordination of learned movement patterns. So these guys make adjustments to the patterns and rhythms. So for example, once you decide to walk and you start walking, the basal nuclei help with the switching from one leg to the other and alternating, one arm being thrown forward and the other being thrown backwards. So sort of the rhythmic and patterns to moving. And anytime you do also make a movement, the basal nuclei plays a role in inhibiting the opposing muscle group. So if you want to flex your arm, then it's going to help to inhibit the extensors of the arm, and etc. So your decisions to make a movement and the start of the movement doesn't start with the basal nuclei, but the basal nuclei help to tailor your movements and to control your movements. So yes, lots of different areas in the brain related to muscle movement. The basal nuclei are also closely linked with the substantia nigra in the midbrain, as we discussed earlier. This is this actual black area in the membrane. These are neurons that use the dopamine as their neurotransmitter. And so this little chart down here, you don't need to memorize this, but this is just to show you that there is a lot of communication between the basal nuclei, which is shown in the purple and the pink. You've got the substantia nigra, which are those dopaminergic neurons in the brainstem. Your thalamus also gets involved with some motor movements, two different types of thalamic nuclei. And then, of course, your cortex, where this is where you're actually going to decide when you want to start a particular movement. So Parkinson's disease is the loss of the dopaminergic neurons. And so when these neurons die, it leads to Parkinson's disease. And Parkinson's disease is considered to be a progressive disorder. So it usually starts out very mild. So not all of the neurons die at once. But as the disease progresses, more and more neurons die, and this leads to an increase in the symptoms. So specifically, the dopamine neurons in the substantia nigra die. And the substantia nigra, again, is that area in the brainstem. And then because the substantia nigra communicates back and forth with the basal nuclei, the death of these neurons means that suddenly the activity of the basal nuclei is now dysregulated. So the basal nuclei can't function like it normally would. So you have difficulty starting movements. So you don't get those opposing muscles to relax, and your muscle tone doesn't shift the way it should. So you have trouble actually starting a movement. And then once you do start a movement, you can have difficulty controlling that movement, and you might actually have to concentrate very hard to produce a controlled movement. And that is actually what we just call the tip of the Parkinson's iceberg. So what people see is they see people that have a tremor, or they see the rigidity. That's when you can't start a movement. They see the bradykinesia, which is very slow movements, or akinesia, which are just, improper type movements, disturbances in the posture, lack of facial expression. But there's actually a lot more going on, and all of this stuff down here in the bottom, this is all related to the loss of those dopamine neurons. Constipation, loss of smell, we actually are learning are two of the most common prequel symptoms that can alert doctors that someone might be developing Parkinson's before they ever start showing any issues with their movement. And as you can see from this list, there's a lot of other things that's also associated with Parkinson's. The risk factors to get Parkinson's is age. So the older you get, the more of a chance you have. And that's because with age, things break down, including your dopamine neurons. And then we've also learned that pesticides, ingestion of pesticides can cause specifically the dopamine neurons to die. Without getting into all the technical details, dopamine neurons are more sensitive to pesticides, and they are the first ones to die. So if you work in an agricultural environment, work on a farm, work around a lot of pesticides, you do have a much higher chance of developing Parkinson's disease. And then we've also identified some genetic mutations that can make you more likely to actually come down with Parkinson's disease. In this section, we'll look at the motor and sensory areas of the cerebrum, as well as the association areas. So first, let's look at the primary motor and sensory areas, starting with the primary motor cortex. So that is shown in the dark red in this image. And as a reminder, this is on the precentral gyrus of the frontal lobe. So the primary motor cortex has neurons for each of your skeletal muscles, and so this is where the instructions to start a movement comes from. So for example, there is a section that's devoted just to your bicep and your arm, and so the neurons that are going to control those movements start in the primary motor cortex. They're going to be adjusted by the basal nuclei and the cerebellum, but this is where the command is going to start. Then we have sensory areas. The primary somatosensory cortex is located on the postcentral gyrus of the parietal lobe, and that is shown in the dark blue here. So this is where the sensations of your general senses, such as touch, pressure, pain, vibration, temperature, proprioception, those signals come directly to the primary somatosensory cortex. They are filtered through the thalamus first, but then this is where they end up. The visual cortex is a specific part of the occipital lobe. So if you look at the occipital lobe back here in green, the visual cortex is the very back of it that's shown in the darker shade of green. This is where your visual information from your eyes is going to go to. So this is going to be the center that receives that input that's coming directly from the eyes. So this is where sight information starts to be processed. The auditory cortex is a specific part of the temporal lobe. So the temporal lobe is shown in yellow. The auditory cortex is the specific part that is in this brighter yellow. And this is going to get nerve signals directly from your ears. So this is where your hearing information is going to travel from your inner ear, and it's going to go here first. Your olfactory cortex is also in the temporal lobe, and it's shown as this sort of light pink. It's kind of like deeper inside the temporal lobe. This is where your sense of smell is processed, and this is the only sense where the neurons do not pass through the thalamus first. And all of these other sensory information pathways, the information will go through the thalamus for relay and filtering. And then there's a gustatory cortex that's shown in this purple color. This is partially on the frontal lobe and partially on the insula. And this is where your taste information from the taste buds on your tongue is going to go. So these are our primary motor and sensory areas. So our primary movements, so where our movements originate, and then this is where all of our sensory information is first going to go to before it's processed and integrated. Okay, so we have that raw data, so the raw movement data and the raw sensory data, but now we have to do sort of higher level functions with it. So we have areas in our brain called association areas, and these coordinate incoming information. This provides interpretation, planning, and deciding what actions to take, so our outgoing sort of motor responses to the incoming data. So these are called association areas. So this is like the next step up on the sort of cognitive ladder. So we've got the raw signals that come in through our sensory neurons. Now we kind of have to think about them, think what they mean, and what to do about it. So the premotor cortex is actually in the area of the frontal lobe that sits right in front of the primary motor cortex. So it is this lighter shade of red here. And this is where you're actually planning your movements. And this is also sometimes called the somatic motor association area. This is where you decide, okay, I'm going to raise my hand. And so this is going to be the area that makes the decision of what movement you want to do, and it is going to pass that information along to the primary motor cortex, which then will send the signal to the muscles in your hand to make it raise up. There is also a visual association area. It's this lighter green color on the occipital lobe. This is the area that's going to take in the raw visual input data and interpret it. So for example, it's easiest to show you this with the visual data, and then you can kind of imagine how this works with the other areas. For example, your eyes are going to just carry information to your brain about color, shape, textures, right? Your raw visual information is just the image that you're seeing here, right? You're not yet thinking about what it is. You're just getting the raw visual data, right? It looks like you filed little protrusions. I see black. I see some brown. You know, I've got a specific shape. Then that raw data goes from the visual cortex to the visual association area, and the visual association area says, oh, that's a glove. Right? So it takes the information that comes from the raw visual input, and it lets you interpret that data into something meaningful. And if all of this fascinates you like it does me, there's a great book I recommend from Dr. Oliver Sacks called The Man Who Mistook His Wife for a Hat. And the name of this book actually comes from a patient who had a stroke that damaged his visual association area. So he could still see stuff, and the stuff that he would see would still go into his brain, but he could no longer interpret what he was seeing. So he would look at his wife, he wouldn't be able to interpret that that visual input was his wife. So at one of his office visits, he went to go pick up his hat to leave, and he actually tried to pick up his wife and put her on his head because he wasn't able to take the visual input and interpret it as that this is his wife and this is his hat. So it's a great book for learning how these different areas of the brain work together to provide us with our normal function. So there's also an auditory association area, which is this lighter yellow shaded region of the temporal lobe. This is going to interpret the auditory input, and again, it's going to take the raw sounds that come in. So you're hearing my voice, and the raw sounds of my voice are going to your auditory cortex, and then your auditory association area are putting the words together and recognizing this as speech and actually hearing the individual words and what they mean. And likewise, you have a somatosensory association area that sits behind the primary somatosensory cortex. And this is, again, is going to let you interpret the data. So you feel something touching your shoulder, and the somatosensory association cortex is going to let you be, oh, that feels like a finger poking me. Oh, that must be my friend next to me poking me because they want to get my attention, right? So again, this is the interpretation of the raw sensory data. And now we're going to look at the integrative areas of the cerebrum, including the language areas, and we'll talk a little bit about aphasia and dyslexia, and we'll also talk about the prefrontal cortex. So we've talked about the primary motor and sensory areas, and then we talked about the association areas. Well, the integrative areas are even higher areas that integrate multiple types of sensory input and use it to make decisions. So in this particular picture here that I grabbed from somewhere else, the darker salmon colored areas again represent the primary cortex, where raw information is coming in, or we're making raw decisions about what to move. Then we have the yellow areas, which are those secondary association areas that we also talked about. And then you've got the association areas, which are kind of these other areas that are left in this light pink color. And this is going to include even bringing in your limbic areas, so making emotional decisions or having an emotional reaction to what's going on around you. So basically, you can think of the integrative areas as sort of summing up and adding together all of the data that came through from the primary and association areas. So let me give you an example. So right now you have raw visual information coming into your primary visual cortex. So your secondary visual association areas are processing that as to what am I seeing. You've also got information coming into your auditory cortex. Then your auditory association areas are saying, what is this that I'm hearing? You've got raw somatosensory info coming into your somatosensory cortex. The somatosensory association areas are saying, what am I touching? And then your integrative areas is taking all of this together and say, oh, I'm in front of my computer listening to an A&P lecture, and I'm holding a pen for taking notes, right? So it's integrating all of this information and putting it all together to sort of form the whole picture. So we're going from raw data to building up what that raw data is, and then what does it all mean? So as part of the association areas, we include specialized language areas. So the first one is Wernicke's area, and that's shown here in light purple. This is partially in the parietal lobe and partially in the temporal lobe. And Wernicke's area receives information from all of the other sensory association areas, and it integrates the sensory information, and it can also coordinate access to visual and auditory memories. Damage to this area can affect an individual's ability to interpret sensory input. Like, you may be able to know that you are seeing words on the screen, that you're hearing my voice, but you can't put it all together and realize that you're sitting there listening to an A&P lecture. There's a specific part of the Wernicke's area that's also associated with your ability to comprehend language. So if that is damaged, then you don't have language comprehension. You don't know what specific words mean, even though you may be able to speak. And the term dyslexia refers to any disorder that affects the comprehension and use of written words. So people with dyslexia can typically have difficulty reading and writing. It doesn't mean that their intellectual functions are in any way compromised. They still have the same intellectual ability as others. They just have difficulty with reading and writing, and it's thought that this has to do with some forms of the sensory processing. So like the letters get mixed up and read backwards, or in the wrong order, and then again that has to do with the integrative areas. Broca's area is shown in yellow on this image, and it's up here in the frontal cortex. Broca's area plays a role in your ability to speak. So damage to this area results in the inability to speak words, although you can still produce sounds, but you can't make specific words. And aphasia is a term that we use to describe any disorder that affects your ability to speak or read. So aphasia could affect either Broca's area or Wernicke's area. There are multiple different types of aphasia. Some people can even have very specific aphasias, like they can't speak names or they can't remember names. But I'm going to go over 3 of the most common. Global aphasia is the worst. With global aphasia, individuals can't speak, read, or understand speech. So that's the worst type of aphasia. Broca aphasia is where individuals can understand others, but they have difficulty talking. So I'm going to show you an example of Broca aphasia that was caused by a stroke that affected Broca's area. So this is a type of aphasia called Broca's aphasia, and this is where you have difficulty actually speaking, although you can understand words spoken to you just fine. This is also called non-fluent aphasia.
Can you tell us your name?
Mike Caputo.
And Mike, when was your stroke?
I was 7 years ago.
Okay. And what did you used to do?
Worked Autodesk, seven sales, and worldwide, and very good. Yeah.
Okay. And who are you looking at over there when you turn your head?
That's my wife.
Okay. And why is she helping you to talk?
She's a speech.
So you have trouble with your speech?
Yeah.
And what's that called?
Aphasia.
All right. And so why don't you work now?
I do.
And what do you do now?
Voices of Hope aphasia.
And what is Voices of Hope?
Peterburg, and Dr. Hinckley, and myself.
So you get the idea. They have normal intelligence and they understand what is being said to them, but they have difficulty actually speaking the words. So that was Broca aphasia. And then another type of aphasia is called Wernicke's aphasia. And with this type of aphasia, individuals can speak, but what they say doesn't make sense. So this is more of a damage to Wernicke's area. So they're still able to speak, but they lose the comprehension of the language and the words. And this is also sometimes called fluent aphasia. And here's an example of that. So this is fluent aphasia, also known as Wernicke's aphasia.
Hi, Byron. How are you?
I'm happy. Are you pretty? You look good.
What are you doing today?
We stayed with the water over here at the moment and talked with the people for them over there. They're diving for them at the moment. They'll save in the moment. He'll have water very soon for him. With luck for him.
So we're on a cruise and we're about to
We
get to
will
June.
store it right here and they'll save their hands right there for them.
And what were we just doing with the iPad?
Right at the moment they don't show a darn thing.
The iPad that we were doing. Like here?
I might change for me and change hands for me. It would happen. I would talk with Donna sometimes. We're out with them. Other people are working with them. I'm very happy with them. This girl was really good and happy when I played golf and hit other.
So they can speak, but they don't have the language comprehension. Most of the words that they say do not make sense. So that was Wernicke's aphasia. And I'm just giving you some examples just because I find the brain fascinating myself. I'm a neuroscientist, and so it fascinates me how we figure out how different areas of the brain do different things. In terms of this course and what you're going to be tested over, I just want you to know the basics of what dyslexia and aphasia are. So the last part of the cerebrum integrative areas we're going to talk about is the prefrontal cortex. So this is the very front of the frontal cortex. So that's why it's called the prefrontal cortex. It's shown in light blue in this image down here. This is where you have your abstract intellectual functions. So this is where you predict the consequences of your actions, where you understand temporal relationships like yesterday, today, and tomorrow. You make executive decisions with all of the information you have. You plan for the future. A lot of people think this is also where the seat of the personality is. So this is like the highest of the high order functions that your brain does is in the prefrontal cortex. And a lot of us have a war going on all the time between our prefrontal cortex and our limbic system. For example, if you are a procrastinator, right, then you're giving in more to your limbic system. Your limbic system says, yeah, let's go watch that TV show because that makes us feel good, you know. So you go and you binge watch some Netflix show, whereas your prefrontal cortex is the one that says, no, we need to be studying. We need to make sure we spend enough time on this material because it's going to help us get an A on the next exam. So a lot of the sort of conflict that a lot of us have with what we want to be doing with what versus what we need to be doing is our prefrontal cortex at odds with our limbic system. And then back in the day, they did prefrontal lobotomies where they would stick an ice pick or other sharp tool either up the nose or through the orbital cavity, and they would basically just scramble the prefrontal cortex. And they used this was an old method used to quote unquote cure those with violent or antisocial behavior, but in a lot of cases it would just kind of make them mindless zombies because then they lost all of these higher level abstract intellectual functions. This section will conclude our talk on the cerebrum, and we're going to look at lateralization and EEGs, and we'll talk about a few things such as disconnection syndrome, seizures and epilepsy, and concussion and CTE. So lateralization is a word that refers to the fact that our two cerebral hemispheres are not entirely equal in function. Although they both have the same lobes and the same general layout of gyri and sulci, each hemisphere also does have its own specific task that it does. For example, the speech center that allows you to talk is only located in the left hemisphere of the brain. So in general, the left side of the brain is specialized for language, speech, as I just mentioned. It's more of your analytical side. So when you're like studying math and science, that's more of your left brain. It's also your more logical and detail-oriented hemisphere. The right side is more attuned to the sensory environment and sensory information. It also has the part of the brain that allows you to recognize faces. It's also good with spatial arrangements, putting things into emotional context, and then intuition and creativity. So sometimes you hear people say like, well, I'm right-brained or left-brained, and that's kind of a simplification of the whole thing because we all need both sides of our brain, and they need to be able to communicate with each other. But that term, I'm right-brained or left-brained, tends to come with the fact that each side of the brain does specialize in different types of processing. So as I mentioned, we all need both sides, and both sides need to communicate with each other. And so the left and right sides communicate by sending axons across to each other, and the area where those axons cross from one side to the other is called the corpus callosum. So the corpus callosum are the white matter. Again, this is the myelinated axons, and so these are the axons, which we also call fibers or tracts, that connect the left and right hemisphere of the brain. Disconnection syndrome is a term that we use to describe any condition or any damage to white matter tracts. And again, remember, white matter tracts are myelinated axons, so it's carrying information from one place to another. So any damage to these tracts can disrupt the communication between different areas of the brain. And so, of course, the result's going to depend on specifically which areas of the brain get separated and can no longer communicate with each other. One specific type is called callosal syndrome, also known as split brain. This is a type of disconnection syndrome that results from a severed corpus callosum, so the two hemispheres can no longer communicate to each other. This is usually done as a surgery for very severe cases of epilepsy. So the people who have had their corpus callosum severed have taught us a lot about the specialization of each hemisphere. And so I'm going to give you an example, and then I'm also posting a video online where you can see this in action. So it's kind of fun to watch. It is an older video, so it is kind of grainy, but it's still on there for you to see. So in general, each hemisphere controls the opposite side of the body. So for example, the left side of the brain controls the muscles on the right side and gets sensory information from the right side. And the right side of the brain controls the muscles on the left side and gets sensory information from the left side. And then when we talk about our visual field, or what we see, each hemisphere of the brain gets visual information from only one half of the visual field. And that is because part of the axons that make up the optic nerve cross over at this structure called the optic chiasm. So part of the fibers from the left eye cross over to go to the left side, part go to the right side, and the same with the right eye. So what essentially happens is, if you imagine a center point when you're looking straight ahead, everything to the left of that point only goes to the right side of the brain, and everything to the right side of that point only goes to the left side of the brain. So this is a case of where your eyes share information, but the brain ends up only seeing either the left visual field or the right visual field, but not both, unless the two hemispheres can communicate. So in a person with callosal syndrome or split brain syndrome, if you show them an image only to their left visual field, so they only see an image here, meaning only their right brain is getting the image info. None of that image info goes to the left brain. So the person will say, I didn't see anything, because the left side, which didn't get to see the image, controls the speech center. So this is the side of the brain that controls you talking. So the person would say, no, I didn't see anything. The right brain saw it, but the right brain can't access the speech center if the corpus callosum is cut. And even more interestingly, if you then put a pencil in the person's left hand, and remember the left hand is controlled by the right side, and the right side saw the image, the person will actually draw what they saw, even though they are verbally telling you, I didn't see anything. And then as they are drawing the image with their left hand, they might suddenly say, oh, it's a cat. And that's because the left side of the brain, which controls the speech and which didn't originally see the image, sees what is being drawn by the left hand and is figuring it out. So it's almost like you can play Pictionary with yourself, because the two halves of your brain can't communicate across the corpus callosum, so they're communicating out on that piece of paper where you're drawing the picture. And again, you can see this in action. There's a split brain video that I'm posting on D2L that you can watch if you want to see more of this. It also, in the video, it shows the experiment they use to show that the right side of the brain is the only side that can recognize faces. So an electroencephalogram, also known as an EEG, is a tool we use to measure the brain's electrical activity. So as we covered in Chapter 12, all of these action potentials are little electrical signals. So all together we can pick them up on equipment that monitors electrical signals. And we commonly call these brain waves. And there are 4 major types of brain waves. Alpha waves are what you see when you're awake but resting, typically with your eyes closed, and they disappear when you go to sleep or when you concentrate on something. So usually once you open your eyes and you're looking around, your brain is focusing on what you're seeing and the alpha waves go away. Beta waves are when you're focused and concentrating on something. You can also see these when you're stressed or tensed, although I would tend to say that when you're stressed or tensed, that's because you're focused or thinking about something that's stressing you out. Theta waves are usually only seen in children and then in intensely frustrated adults. And then delta waves are seen during sleep, also in some pathological conditions like with certain types of tumors, but this is typically what you would see during slow wave sleep. So a seizure is when you have excessive synchronous neural activity in the brain. So basically you've got too much excitatory input and not enough inhibitory input, and all of the neurons start overfiring, firing too many action potentials, and they start all firing at the same time so that it's synchronized. And the effects of a seizure depend on the location within the brain and then how widespread this abnormal electrical activity is. So for example, two broad types of seizures, you can divide them into partial seizures in which the abnormal electrical activity is only in a small portion of the brain and the EEG for the remainder of the brain looks normal. So you can actually only see the seizure activity in a specific part of the brain, versus a generalized seizure. This is the ones that we call like the grand mal or tonic-clonic seizures. These occur where the seizures activity spreads over the entire brain, so you would see it all across the EEG. Epilepsy is actually a diagnosis you get only after you've had 2 or more unprovoked seizures. And that means that no other cause for those seizures could be found. Like it wasn't a seizure due to a fever or poisoning or a recent head injury. If you just have a seizure out of the blue and then you have another one out of the blue, you'll end up with a diagnosis of epilepsy. We don't know why a lot of people have epilepsy. This is actually what I studied in grad school. We do know of some genetic mutations that can lead to them, and that's what I focused on, was genetic causes of epilepsy. But a good number of epilepsy cases, we still don't know the exact cause as to why the person has this random seizure activity. And I want to put this out there because I know a lot of you have kids or will have kids. Febrile seizures, which is a seizure that is triggered by a fever. So the word febrile is a prefix that means fever related. So febrile seizures are actually fairly common in kids between the ages of 6 months old and 5 years old. And again, these seizures are triggered by a fever, and they're not anything to be too concerned about unless your kid has multiple of these, but 1 or 2 of these is generally nothing to worry about, and they're common in kids actually. So the last little section here will be on concussions and traumatic brain injury. So a traumatic brain injury, of course, is anything that's going to damage the brain, and a concussion is the most common type of TBI. And so with a concussion, the brain is hitting up against the inside of the cranial bones. So you can see in this little GIF here, the brain's consistency is kind of got like a jello mold kind of consistency. And so if you do have a hard hit to the head, it can cause the brain to jump around within the cranial cavity. You do have cerebrospinal fluid and your meninges, which is supposed to help keep the brain in place. But even with a hard hit to the head, it still can hit up against the inside of the cranial bones. Concussion symptoms can appear right away, or sometimes they may not even appear for a day or 2. So you should take any hit to the head seriously. Even more dangerous than a concussion is a subdural or epidural hemorrhage. So it's generally a good idea to have any type of head injury checked out as soon as possible. So what happens with the concussion is after the brain hits up against the inside of the skull, you get inflammatory markers, microglia go to that area. You might have a dysregulation of the ion concentration, which can lead to excitotoxicity, which are the neurons that fire too much and then eventually die because they are firing too much. And then you also may even get stretching or eventually tearing of the axons, which will affect your white matter, which is your myelinated axons. And I have a quick little video, again, just to drive home the point that a concussion has to do when your brain hits up against the inside of your cranial bones.
A concussion is a type of traumatic brain injury, or TBI, caused by a bump, blow, or jolt to the head, or by a hit to the body that causes your head and brain to move rapidly back and forth. This sudden movement can literally cause the brain to bounce around or twist in the skull, stretching and damaging the brain cells and creating chemical changes in the brain. What you might not know is that these chemical changes make the brain more sensitive to any increased stress or injury until it fully recovers.
So as he mentioned right there at the end of that video, repeated concussions are even more dangerous than just having one concussion over your lifespan. And if you haven't seen the movie Concussion with Will Smith, I recommend you watch it. It's actually a true story about the doctor who first started trying to make more widely known the science behind repeated concussions. So chronic traumatic encephalopathy, or CTE, is the progressive loss of brain function due to repeated concussions. And we have seen CTE show up in boxers, American football players, and soccer players who frequently head the ball, which is they hit the soccer ball with their head instead of their feet. And so chronic traumatic encephalopathy, here is an image from a scientific paper. So at the top you see a normal brain, and at the bottom you see the brain of a retired professional football player. And you can see that the repeated concussions, and especially the repeated concussions that occur before a prior concussion has healed, dramatically reduces both the gray matter and the white matter within the brain. So the brain actually, the cells die and the brain matter shrinks. So again, if a second injury happens before the first injury has had time to heal, you're going to get even worse results that are going to cause more rapid progression of the disease. In this last section, we'll be looking at the cranial nerves. So these are 12 peripheral nerves that connect directly to the brain. The other peripheral nerves connect to the spinal cord and will be covered in later chapters. We'll also talk about the pathophysiology behind migraine in this section. So there are 12 pairs of cranial nerves, and we usually use Roman numerals to differentiate between the cranial nerves. And each cranial nerve is classified as either carrying only sensory information, carrying only motor information, or a mixed nerve, meaning it carries sensory and motor information. And you only need to know the details that are covered in this PowerPoint. I'm not going over all of the details that are in the chapter. Cranial nerve number 1, also called CN1, is a sensory-only cranial nerve. It's called the olfactory nerve, and so this is going to carry your sense of smell. So the olfactory nerve itself is actually in the roof of the nasal cavity. So these tiny little nerves right here, these are actually the olfactory nerves, cranial nerve number 1. And they synapse with a structure called the olfactory bulb, which sits right above the roof of the nose. And then the axons of the olfactory bulb neurons become the olfactory tract, which carry the information back to the olfactory cortex of the temporal lobe. And so in lab, you can see the olfactory bulb underneath the frontal lobes and the olfactory tract running behind it. Cranial nerve number 2, or CN2, is the optic nerve. This is also a sensory-only cranial nerve, and it carries your visual information. So you have an optic nerve running back from each eye. And then some of the fibers from the optic nerve cross sides at the optic chiasm, as we talked before when we introduced you to lateralization. So some of the fibers from the left eye go to the right side of the brain, and some of the fibers from the right eye go to the left side of the brain. And then after these fibers cross at the optic chiasm, we then call the portion behind that the optic tract. The next 3 cranial nerves we're going to lump together. All 3 of these are motor-only cranial nerves that control muscle movement, that moves your eyeball. So we call these extrinsic eye muscles, and we're going to talk more about these in A and P2, but we're introducing them to you now. There's the oculomotor nerve, which is cranial nerve number 3. This controls 4 of the 6 extrinsic eye muscles. There's the trochlear nerve, cranial nerve number 4. And then we're jumping to number 6, the abducens nerve, cranial nerve number 6. The trochlear nerve and the abducens nerve each control 1 of the extrinsic eye muscles. And again, these are the muscles that actually move the eyeball that allow you to look up, down, and left and right. So the nerve we just skipped over and we're going back to is cranial nerve number 5. This is the trigeminal nerve. This is actually the largest of the 12 cranial nerves. The trigeminal nerve is a mixed nerve, meaning it carries both sensory and motor information. It has 3 major divisions. That's where the prefix tri comes from. So it has an upper ophthalmic branch, then it has a middle maxillary branch and a lower mandibular branch. So as you can see, it's quite widespread in the areas of the face that it innervates. And one important thing to know about the trigeminal nerve is that it's also involved in migraine pathophysiology. So we know that the trigeminal nerves, they actually wrap around blood vessels that are in the face and cranium. And as part of the nerve contact with these blood vessels, you get release of certain inflammatory chemicals that travel along the trigeminal nerve to the brainstem, which then feed back into the cortex, and you end up with a migraine, which is a really strong, powerful headache that can last as long as 72 hours without treatment. And then with the effects in the cortex, some people can have auras, which mean they see like lights and stuff right before they get a headache. They can be highly sensitive to sounds, which is phonophobia, highly sensitive to light, which is photophobia. And so we definitely know that the trigeminal nerve does play a key role in the way that migraine works. And a lot of the newer migraine medicines that are just now hitting the market in the last 5 years or so are actually targeting the inflammatory markers that the trigeminal nerve interacts with alongside the blood vessels. Nerve number 7 is the facial nerve, so cranial nerve VII. This is also a mixed nerve, meaning it carries both sensory and motor information. It has quite a few branches, as you can see from the picture on the right that shows the 5 different branches and the areas of the face and neck that each branch is going to affect. The only important thing that I'm going to let you know about this nerve now is that inflammation of this nerve can cause a condition called Bell's palsy. And this results in the paralysis of facial muscles. Usually you see it when a person has like a droopy eye or a droop to one side of their mouth. And you can also lose some of your taste sensation since the facial nerve carries quite a bit of the sensory information from the tongue. Cranial nerve number 8 is the vestibulocochlear nerve. This is a sensory-only cranial nerve, so it's only carrying sensory information. This nerve carries hearing information as well as your balance and equilibrium info. So there are 2 main branches. You have a vestibular branch, which is responsible for carrying information about your equilibrium. And then you have a cochlear branch, which is responsible for carrying the hearing information. Cranial nerve number 9 is the glossopharyngeal nerve. This is a mixed nerve, meaning it carries sensory and motor information. It innervates the tongue, the pharynx, and the carotid arteries. And in fact, glosso is a prefix that means tongue, and then pharyngeal means pharynx. So the name tells you that this nerve is going to innervate both the tongue and the pharynx. Cranial nerve number 10 is the vagus nerve. This is the longest cranial nerve. So it starts up at the brainstem, and this one travels all the way down throughout the thorax and throughout the abdomen, and it makes contact with many of the thoracic organs as well as the abdominopelvic organs. So the vagus nerve is a sensory and motor nerve, so it's mixed. As I just said, it's widely distributed in the thorax and abdomen. And in fact, the visceral sensory information that it picks up, and again, visceral means organ, so that means the sensory information it picks up from your organs is vital to the autonomic control of the visceral functions, which we're going to talk about in chapter 16. So the vagus nerve is the only cranial nerve that actually leaves the head and neck region and goes down into the main torso of the body. Vagal nerve stimulation, or VNS, is actually a relatively newish procedure that is being used to treat both epilepsy and depression. However, it is also being looked into for treating other disorders. There's some ongoing studies for treating arthritis and for treating inflammatory bowel disease. And if you want to read more about vagal nerve stimulation and how it can help all of these different types of disorders, I'm posting an article on D2L that says what happens in Vegas does not stay in Vegas. Cranial nerve number 11 is the accessory nerve. This is a motor-only nerve, and it controls muscles of the neck and the upper back. And last but not least, the hypoglossal nerve, cranial nerve number 12, is a motor-only nerve, and this one controls just tongue movements. And again, remember, glossal or glosso is a root word that means tongue. And then here is a summary of the 12 cranial nerves. And again, you should know their names, their numbers, whether they're sensory, motor, or mixed, and then what other information that I presented on the slides. And that is the end of chapter 14.